Hello, welcome back. So there is a disturbance earlier. So this is a continuation of the previous class. So DNA damage checkpoints. So we are in DNA damage checkpoints, ATM and ATR, how they are activating. They are activated by the double standard DNA breaks here. ATR is activated by the single standard or unreplicated DNA. Once activated by DNA damage, this ATM or ATR phosphorylate and activate the checkpoint kinases. CHK means checkpoint kinases. There are two, CHK1 and CHK2. They will induce cell cycle arrest by phosphorylating or inducing the degradation of CDC25 phosphatases. So in this manner, if there is a detection of double standard breaks or single standard breaks, then these kinases via ATM and ATR phosphorylated and activated, and they will arrest the cell cycle. Next, what is the role of P53 in G1 arrest? So this is very important molecule, P53. So if there is a DNA damage, P53 levels will increase. In mammalian cells, cell arrest at G1 checkpoint is mediated by a protein called P53. P53 is phosphorylated by both ATM and CHK2. P53's transcription factor, its increased expression leads to the induction of CYP or KIP family CDK inhibitors, which will further activate P21. This P21 protein inhibits CDK2 or cyclin E complexes, leading to cell cycle arrest in G1. The P53 molecule regulation is this. Next, coming to the mitosis. The mitosis phase, it occurs during mitosis. It is having five things are going to happen in mitosis phase. The first is the chromosome condenses. The second thing is nuclear envelope breaks down. The third thing is cytoskeleton reorganizes to form the mitotic spindle. The fourth one, chromosomes move to opposite poles. Fifth one is chromosome segregation followed by cytokinesis. Now we will see one by one. M phase is divided into four phases. The first one is prophase, metaphase, second one, third one, anaphase, fourth one, telophase. It is followed by cytokinesis. Prophase. What is happens in the prophase? In the beginning of prophase, condensed chromosomes. So you can see in the picture, the chromosomes are condensed. And you can see two sister chromatids remain intertwined throughout the G2 and S during chromatin condensation. So these are the two chromosomes which sister chromatids you can see in the prophase. And also development of mitotic spindle initiated during prophase. You can see the spindles are going to start on the left and right side. These are centrosomes. So they are going to produce mitotic spindles. That centrosomes duplicated during the interphase, they separate and move towards the opposite sides of the nucleus. You can see the two. You can see the two centrosomes here. Two poles of the mitotic spindle begins during late prophase. The spindle fibers will be produced after late prophase. Next, I'll go to prometaphase and metaphase. In prometaphase, the microtubules of mitotic spindle attach to the kinetochore of the condensed chromosomes. You can see in the picture, these are the chromosomes. Chromosomes are separated in its structure as sister chromatids. The center part is called centromere. To the centromere, kinetochore is a thing that where microtubules will come and attach. So kinetochores of sister chromatids are oriented on opposite sides of the chromosomes. Chromosomes attach to the microtubules emanating from the opposite poles of the spindle. So you can see the spindle fibers are coming and they will attach to the kinetochores. In this metaphase, chromosomes shuffle back and forth and align on the metaphase plate in the middle of the cell, the center of the spindle. This is about metaphase. And when you go into anaphase breakage of the link between sister chromatids, they will be getting separated and move to the opposite poles of the spindle. So you can see in the picture, they are pulled by the microtubules. Two sister chromatids are pulled separately and move to the opposite poles. Telophase. Telophase where nuclear reforms and the chromosomes decondense. So this is happening after the completion of the anaphase. And next it is followed by cytokinesis. Cytokinesis begins during the late anaphase, almost completes at the end of the telophase. So here you can see the two cells are separated, which are having nucleus. So this is called cytokinesis. Overall, mitotic phase or mitosis is having prophase, metaphase, anaphase, telophase, and it is followed by cytokinesis. Finally, the cells are divided. Here we'll see about regulation of M phase, how it is regulated. CDK1 or cyclin B, which is also called NPF, earlier we have discussed. This acts as a master regulator of the M phase transition, both by activating other mitotic protein kinases and by directly phosphorylating some of the structural proteins involved in the cellular organization. Chromatin condensation is driven by protein complexes. Two complexes are there. They are condensins and cohesins. So these condensins and cohesins play a very important role in chromosome condensation and separation. So here they are members of SMC. SMC means structural maintenance of chromatin. And we see in the next slide how these cohesins and condensins work. So you can see in the picture condensins. Condensins which are in a green color. So what they will do means they will condense the chromosomes. They will condense the chromosomes. You can see here these green parts, they will condense the chromosomes. And you can see in the middle, this is here we are having cohesins. We are also having centromere. So this is sister chromatid, but before the formation of sister chromatids, these two sister chromatids are attached by these cohesins. When they are entering into the M phase, then these cohesins are replaced by condensins. So now you'll see proteins, cohesins bind to the DNA in S phase and maintain the linkage between sister chromatids following DNA replication. See, these cohesins are binded to the DNA in S phase and they will be continued until the completion of the replication. Next, as a cell enters M phase, condensins activated by these condensins are activated by CDK1 or cyclin D, means MPF factor phosphorylation. So these condensins again replaced by the cohesins along the most of the length of the chromosomes and only linked at the centromere. So like this, the MPF factor or CDK1 or cyclin B regulators regulate the M phase to when to enter into the next phase and where to stop. Okay, so this is about cell cycle regulation. Thank you, and I'll come with other classes next week. Goodbye.